Is it started?
Not quite, not yet.
Okay.
Okay. It just sits there spinning for a little while and then gives a short countdown.
Okay.
Yeah. Okay. And in 3, 2, 1, and live.
Okay. Hi, I'm Jeremy from the Riffle Lab at the University of Washington. Thank you for attending my talk on how atmospheric oxidation impacts odor navigation by hawkmoth pollinators. There are many open questions on how air pollution affects plant pollinator communities. My research focuses on a primrose hawkmoth plant pollinator system. And the questions that I wish to answer are, what scent chemicals are important for attracting hawkmoth pollinators? How nighttime atmospheric oxidation degrades scent chemicals in the air? And how this degradation impacts hawkmoth navigation? And finally, how this impacts floral visitations by hawkmoths in the field. So previous research has shown that ozone reacts with floral scent, resulting in decreased attractiveness to pollinators. So this experiment by Faria Armengol et al. In 2015, of which James Bland is one of the authors, used a very similar experimental setup to me. So what they did was they mixed a scent source with an oxidant and reacted them in glass flow tubes. So according to a modeling study by McFrederick et al. In 2018, atmospheric oxidation has drastically reduced the transmission of some floral scent compounds in the atmosphere compared to pre-industrial times. For example, the transmission of ocimene, which is a common floral scent compound, has been modeled to be reduced from over 80% over a kilometer under pre-industrial conditions to as little as 0% over a kilometer under more polluted present-day pollution. So reactive oxygen species, or ROS, like ozone and nitrogen oxides, degrade scent compounds in the atmosphere. Hydroxy radicals, or OH, and nitrate radicals, or NO3, as well as ozone, or O3, are the three main types of ROS that degrade chemicals in the atmosphere. The different ROSs have different reactivities towards volatiles, with the hydroxy and nitrate radicals being significantly more reactive than ozone towards many volatiles. So nitrate radicals are the dominant nighttime radical source for atmospheric oxidation, and it is formed from the reaction of ozone with nitrogen oxides, which are largely from human pollution in the atmosphere. And in the daytime, sunlight rapidly destroys nitrate radicals, and hydroxy radicals are formed from the reaction of ozone with sunlight and water vapor instead. And these hydroxy radicals are the dominant daytime radical source for atmospheric oxidation. My study plant is the pale evening primrose, Unathera pallida, which grows in the desert sands of eastern Washington. So it's a valuable resource for pollinators of that arid region. And Unathera pallida is pollinated by, visited by a rich diversity of pollinators, including diurnal butterflies, various species of bees, syrphid flies, as well as like crepuscular lasioglossum bees, various species of hawk moths, some smaller moths, and even some more unusual visitors like these mammaleontids. And we are studying how atmospheric pollution impacts the reproductive success of Unathera pallida by affecting pollinator visitation rates. So Hyles lineata and some Manduca species are the dominant nocturnal pollinators of Unathera pallida in the field. And we are using Manduca sexta as a proxy for the field Manduca species in our lab experiments. So my experimental objectives are to determine what scent chemicals in Unathera pallida scent the hawkmoths respond to, and how they are degraded by nocturnal atmospheric oxidation, how this scent degradation affects hawkmoth navigation, and finally, how this impacts floral visitations by hawk moths in the field. So the first thing I did was I collected Oenothera pallida floral scent samples using dynamic sorption by using a pump to draw scented air from bagged Oenothera pallida flowers over these Poropac scent traps. Next, I analyzed the scent samples using GCMS to determine the chemical composition of the scent, and GCEAD to determine which of the scent compounds are antennographically active with Hyles lineata and Manduca sexta antennae. And then finally, I created a scent blend of these antennographically active compounds. So from the GCEAD data of Hyles lineata, which is shown above, and Manduca sexta shown below, I created a scent blend of 12 antennographically active compounds. And you can see that Manduca sexta below responds to actually a subset of the compounds that Hyles lineata responds to. And we use a chemical ionization time of flight mass spectrometer for online analysis of the simulated atmospheric degradation of floral scent through a gas flow tube. And we used different chemical ionization schemes, including benzene cation chemical ionization for the detection of the aromatics in a scent. And we use Volker's PTR, proton transfer reaction chemical ionization, for the detection of other chemicals. And here's kind of a schematic of the Volker's PTR that was developed by Torfwerk and Aerodyne. So different floral scent compounds react differently in the presence of atmospheric oxidants. For example, methyl salicylate and osmine are reactive towards these oxidants, and methyl salicylate reacted to form an oxide in our experiments as seen in this mass spectrogram. So whereas other compounds like eucalyptol are relatively unreactive towards ozone and nitrate radicals, as you may be able to deduce from its kind of saturated structure. So different floral scent compounds are also differentially affected by ozone versus nitrate radical oxidation. So monoterpenes are the most severely affected, which is consistent with a lot of the existing literature out there. While some of the other compounds like the butanol oxime are not significantly affected by ozone or nitrate radical oxidation. But the combination of ozone and nitrate radicals degrades monoterpenes significantly more than ozone alone does. So next we conducted no-choice behavioral assays in a wind tunnel towards these floral scent sources in order to determine the proportions of moths which would successfully locate the scent sources by flying upwind. And here we see a Manduca sexta successfully navigating to the scent source and landing on the filter paper cone of the scent source. But this, however, was not a feeding visit, as its proboscis was not extended. And in our trials, only visits with an extended proboscis were scored as successful visits. Next, we compared the moth responses towards my floral scent blend and scent blend that was oxidized with ozone and nitrate radicals. And the results show that oxidation significantly reduced Hyles lineata response towards my scent blend, while Manduca sexta response was unaffected. And then we proceeded to test the effects of scent dilution on Manduca sexta behavioral responses. And we found that Manduca sexta responds robustly even to 1,000 times diluted scent blend, although that started to be statistically different from the undiluted scent blend. So finally, we compared the effects of oxidation of undiluted versus diluted scent blend. And we found that nitrate radical oxidation of my diluted scent blend yielded behaviorally significant results, whereas oxidation by ozone alone did not yield significant results. And so this suggests that while Manduka sexta foraging behavior seems to be more resistant to the impacts of atmospheric oxidation than with Hyles lineata, they might be susceptible to these impacts further downwind with scent dispersal and dilution. And so in conclusion, we have shown that floral scent chemicals are differently affected by oxidation, that different pollinators are differentially impacted by floral scent oxidation, and that the observed impacts from oxidation to Manduka sexta visitation might be due to an altered ratio of scent components rather than a simple reduction in scent concentration. And so next we plan to investigate how altered scent blend compositions affect antennographic response and also wind tunnel responses, and perhaps see if this impacts visitation rates in semi-field or field settings. And so to do this, we intend to compare wild hawk moth visitation rates to the scent blend versus the oxidized scent blend in the field. However, so far we have had a low visitation rates of moths to our field setup. And here we see a Hyles lineata approaching the filter paper cone of the scent source in the field, but not actually visiting it. So yeah, I've been trying to tweak a few things like introduce humidity, and it's still an ongoing process to try and get them to visit my field setup. So yeah, we would like to acknowledge our funding sources, including various national and local institutions, generous donors who funded my research with awards, and BNG Farms for allowing me access to their land on which my field site is located. And I would also like to thank members of the Riffle and Thornton labs and members of my committee for their valuable help and advice. And yeah, I thank you all for listening to my talk, and I'd love to answer any questions that you might have.
Thank you, Jeremy, very much for your talk on the nighttime interactions. I always have to activate this question chat. But at the moment, no, there is one question coming in.
Okay, yeah.
It's from Silke Alman. Hi, Jeremy. Thanks for your talk. Is it possible that Manduka uses primarily volatiles that are less prone to floral scent oxidation for foraging?
That might be the case, although, yeah, I guess with what I have so far, I'm not sure if I can verify that that's the case. But in the next step, I plan to create scent blends representative of the oxidized scent, and maybe actually maybe not, because if I went back and I looked at the antennograms, sorry, oh there. Yeah, it doesn't, I guess it's hard to tell. It doesn't look like the kind of responses to the particular odorants that are less affected by degradation are larger, but then again, I don't know if the amplitude of the antennal response corresponds with detection by the moth. So yeah, I might need to do like some wind tunnel behavioral tests to see if that's the case. But yeah, that's definitely an interesting thing to consider.
Okay, thank you.
Yeah.
James, do you have a question?
Yeah, I could maybe ask a question. I'm just, it's more a technical question than anything, but nitrate radicals are quite technically tricky to work with.
They are, yes.
So how are you managing with that? I mean,
yeah, so I think the primary thing with that is to keep the setup extremely dry. So here, this is the nitrate radical generated as a tank of diluted nitrogen dioxide, and I have an ozone generator, and there is a 1-inch diameter Teflon tube that I use to react the ozone with the radicals. But yeah, I need to use 0 air for this reaction because any moisture immediately converts the nitrate radicals to nitric acid. And yeah, so that's how I've been doing it. And I've also done, yeah, I use kind of, I think 60 parts per billion of nitrogen dioxide and maybe 120 parts per billion of ozone to keep it kind of atmospherically relevant. And I did do some kind of box modeling of the dynamics of the ozone and nitrogen dioxide to see that I'm getting like a decent amount of nitrate radical formation. Yeah.
Okay, so I think very nice efforts, because I know that it's very tricky to work with, and I haven't really dared to try that myself. So
yeah, it is a pain. And I was actually also trying to investigate the hydroxy radical chemistry with the daytime pollinators, but yeah, I mean, within the span of a PhD, it's like the nitrate radicals itself was challenging enough to deal with.
Sure.
There's one more question now from Birgit. She's asking, do the radicals appear in different concentration through the year?
That's an interesting question. I mean, I don't have direct verification of this because I could measure the actual radical concentrations if I left the mass spectrometer out in the field, but yeah, I haven't done that. So maybe the best effort that I can use to ascertain that is by looking at maybe kind of national maps by the Environmental Protection Agency or various air quality agencies to monitor the levels of ozone and nitrogen dioxide throughout the year. And then I can use kind of like simple box modeling to figure out what these levels are. But yeah, I haven't done that, although at my field site, it's pretty far away from other human activities. So the nitrogen dioxide levels are actually quite low. Even in the summer, it's like less than a part. Yeah, 1 to 2 parts per billion. So I imagine actually at my field site, they are quite low throughout the year. As for hydroxy radicals, that's a bit harder. I think you could model that by modeling kind of the solar irradiance, radiation irradiance with ozone levels and also relative humidity. But yeah, I don't know. I imagine that ozone levels would be low in the winter when there's like less sunlight to generate it from the biogenic VOCs. But yeah, that is an interesting question. But then again, these flowers bloom in the summer, and in the winter it's like really cold out in the desert here, and hardly anything is blooming, and there are hardly any pollinators about. So yeah, it's an interesting question.
Good, thank you.
Yeah.
So there seem to be at the moment no more questions
Okay.
for you, Jeremy. Thank you again for your